Major strategies for gene transfer to plants. Now we have discussed that plants can be reproduced or regenerated from variety of plant tissues like callus, like somatic embryos, from root tips, shoot tips, from leaf part under in vitro conditions. And all these procedures, they provide us an opportunity where we can genetically alter the plants at a particular developmental level. Although it is nowadays it is easy that we can transform the plants so that we can get a transgenic plants with our desirable characteristic features. But the selection of transgenic plants, it is the main challenge that we have to screen like the bacteria that whether plants they have been genetically modified or not. And for this purpose, different strategies have also been developed. So once the plants they have been genetically modified, then we can expect that in the near future that they will produce some valuable commercial products like proteins having chemotherapeutic importance, and even plant cells they can act as the bioreactors to produce different valuable metabolites. So after having an overview of the plant tissue culture techniques that can be exploited for genetic modification, briefly we can focus that what are the major strategies that can be used for plant transformation. स्टार्ट में हमने ये डिस्कस किया कि कुछ बैक्टीरियल पैथोजन ऐसे होते हैं जो कि जेनेटिक मटेरियल को या अपने जेनेटिक मटेरियल को या डीएनए को वो प्लांट में इंट्रोड्यूस कर सकते हैं. तो इसमें जो ज़्यादा माइक्रोब्स यूज़ हुए हैं इट बिलोंग्स टू द जीनस एक्रोबैक्टीरियम. Agrobacterium, it can transfer our gene of interest, so it can be used as a vector. But in addition to that, some other techniques like the direct DNA transfer technologies have also been introduced. And direct DNA transfer technologies, they are mainly based on chemical or physical genetic manipulation so that DNA, without the need of any vector, it can be transferred into the plant genome. Although agrobacterium, it is mainly used for the genetic transformation of dicotyledonous plants, and for monocots, it is not so effective. The reason we will explain later on. And for monocots, some other direct methods have been used, like the particle bombardment, that we can use small metal particles to which our DNA has been attached. And by using shotgun, we can penetrate these small particles into the nuclei of the cells so that DNA is delivered. But like animal cells, some direct methods have also been used that are based on some chemical transformation of the target cells. And here, the calcium phosphate transfection has been used. Like in case the animal cells, protoplasts, they are also cell wall-less, and this calcium phosphate transfection, it is very effective for genetic transformation. Although viruses, they can also be used to deliver our target DNA, but for plants, viruses that can integrate the foreign DNA into the host genome during the infectious cycle has not been reported. But some viral particles, they can be used as an episomal vectors. Episomal Vectors are those vectors that have the ability to replicate either in integrated form or, most importantly, as an autonomous structure. So, in case of episomal vectors, then systemic infection is there in the plants. A number of plant tissues, they may be infected with episomal vectors that can be expressed there and produce our target proteins. So these are a few methods that can be used for plant tissue transformation that are based on bacterial gene delivery system, viral transduction, chemical or physical methods for direct DNA transfer.